So this is a slideshow that's going to introduce you to the technique that's used in throughout really biology and beyond, which is called the polymerase chain reaction, or for short, PCR. And we're not going to go into a lot of details right now in terms of what the applications are for PCR. You'll see some of them as you move through the class. You'll see some of the applications as you move through the lab. But the reason that we're introducing PCR to you right now is because it really is a type of technique that relies upon the basic premises that are found in the study of DNA replication. The difference between PCR and DNA replication is that DNA replication happens within a living cell, and PCR is something that we do in vitro, or that's Latin for in glass, or really in plastic, because we typically do these reactions in small plastic tubes. So when you're thinking about learning about PCR, you're going to be relying upon some of the basic information that you already know about DNA structure, nucleotide structure, and the basic way that DNA polymerases work, which is by adding nucleotides onto a free 3' end of a primer or a short sequence that is adjacent to a template strand. So we're going to step you through kind of an example of how PCR is done, but in this first slide here, we kind of have an overview. So let's kind of start with an overview, and then we'll hone in on each one of the steps in a little more detail later. PCR, the reaction starts with a piece of DNA that you want to amplify, and this is often referred to as your template or sometimes as your target DNA. You put that piece of DNA into a tube, and in that tube are primers which you have designed to have a particular sequence. There are also the deoxynucleotides, which are going to be used to build new DNA. And there's also a polymerase that is added as well, which is going to synthesize the DNA. Unlike DNA replication that relies upon a helicase to separate the double strands, PCR utilizes heat. Remember that when we talked about DNA structure, we talked about the fact that if you add heat to a double-stranded molecule of DNA, those two strands will separate into two single strands. So that is referred to as denaturation, so the two strands separating. The temperature is then lowered a little bit so that primers can find their target and bind to the template. And once these primers bind, they can then be the substrate for a DNA polymerase, which can recognize that free 3' end and extend the primer so that you're generating more DNA sequence. PCR is a series of cycles. Typically, when you do PCR in your reaction, you'll do about 20, 30, or 40 cycles. And with each one of these cycles, you're increasing by twofold the number of DNA fragments that you have in your reaction. So that's why we can think about this as a doubling reaction, and the mathematical equation that would go along with that would be 2 to the n, where n represents the number of cycles. This is a really important technique, as you'll find as you move through your study of biology, and it's a technique that was recognized as being important many years ago, and in fact was the basis for the awarding of a Nobel Prize. So we're going to start with a little more detailed explanation of what happens in PCR so that you can really understand the details of what is occurring in a PCR reaction. So here I've drawn for you a piece of DNA, and I'm telling you that this is a piece of DNA that we would like to amplify. Here the word amplify means make more copies of, increase the number of. And PCR is typically referred to as an amplification process. So in this example, we do not want to amplify or copy this entire sequence. Instead, we want to copy a subset of this sequence, which is highlighted here for you in yellow. And this is a very typical way in which PCR is conceptualized. Let's say that you're doing PCR on a human DNA sample. You do not want to copy all 3 billion nucleotides of the human genome. Instead, you would want to copy a very short fragment. The amount of DNA that can typically be copied in a PCR reaction is something on the order of hundreds to maybe a couple thousand nucleotides, and that's about it. That's the limit of this particular technique. So for our illustration here, we have a larger piece of DNA and we're interested in copying the yellow part that we see here. So if we start with this piece of DNA, as I mentioned before, the first step in PCR is to denature or to separate the strands of the double-stranded DNA from each other. And that is done by application of heat. Typically around 95 degrees Celsius, that's sufficient to separate the two single strands from each other and maintain them as being separate and distinct, not re-annealing to each other to make a double-stranded DNA. As I mentioned before, in this reaction, there are also these primers that are present. And one of the differences between the term primer that you learned when you were learning about DNA replication and the term primer that we're going to use here is that in the cell, when DNA replication is actually happening, the primers are synthesized by an enzyme that's called primase. And primase makes primers that are RNA primers. When we are doing PCR in a test tube, the primers that we are adding to our reaction are primers that are synthesized by a machine, and the sequence of those primers is dictated by the desires of the researcher for the particular application that they are using PCR to achieve. Primers that are typically used for PCR reactions are not made of RNA nucleotides, ribonucleotides. Instead, they are made of deoxyribonucleotides. So these primers are DNA primers. And there are technical reasons for that, but it is one difference that I want to point out between how this happens in the cell and how it happens in a PCR reaction. So these primers are present while the heating of the double-stranded DNA is occurring. And the primers, once they are able to access the single-stranded DNA, and with a slight decrease of temperature, what we'll see is that these primers will bind to or anneal to particular locations within the template DNA. And as you'll see here, the primer that is on the left side of the screen, 5' GATTT CCC, that primer is complementary to the sequence that is present on the bottom of the two strands of the double-stranded DNA. And the other primer is complementary to the top strand. And I want you to really think about how the anti-parallel nature of DNA is important here to really understand how these primers are designed so that they are complementary to this specific sequence. So once those primers recognize their complementary sequence in the template. They will anneal through the hydrogen bonding that occurs between nucleic acids. In order to facilitate this happening, the temperature of the reaction is lowered to around 60 degrees. And there are ways to kind of maximize the efficiency of PCR reactions based upon getting that temperature exactly right, but typically this is around 60 degrees. Under these conditions, the primers will bind to the template, but the two template strands are not able to re-anneal completely to each other. So we have now done the denaturation phase where the template strands separate and the annealing phase where the primers anneal to the templates. And we imagine what it looks like when a DNA polymerase has bound to that 3' end and has started the process of extending or adding nucleotides to that primer. We get an image that looks something like this. So here the primer sequence is in red and the new DNA sequence is in black. And we can see that the DNA polymerase, which is in blue here and is labeled TAQ, because that's its name, TAC polymerase, is adding nucleotides onto the free 3' end. And the DNA polymerase will continue to add nucleotides onto that free 3' end until it gets to the end of the sequence. So it's going to chug along, adding nucleotides, and then when it gets to the end of the sequence, it will simply release the template, and because this is an enzyme that's catalytic, it can go and be involved in another round of DNA synthesis later on. So here we have the end of the first cycle of PCR. We started with one copy of double-stranded DNA, and now we have 2 copies of that double-stranded DNA. Now, the word copy here means that we have twice as many as we started with, but note that neither one of them look exactly like what we started with. Neither one of them looks exactly like the double-stranded template, and neither one of these copies look exactly like what we are trying to do. We are trying to make just a copy of that yellow region. So while we have made more DNA, we haven't made exactly the fragment of DNA that is our intention. After the first cycle, we end up with the 2 products that are shown here, and then the cycle continues. So we move into the second cycle, which involves, once again, heating the samples to 95 degrees so that we have the 2 strands separating or denaturing. Here is what it looks like when that top molecule separates into 2 single strands. And here's what it looks like when that bottom molecule separates into 2 strands. In this reaction, we also have primers. We're going to lower the temperature to 60 degrees so that the primers will bind again. There we see the primers binding to their target sequences in the template strand. The DNA polymerase is still there, so the DNA polymerase is going to recognize those free 3' ends and extend all 4 of these primers so that we end up with reaction products that look something like this. This then is the end of the second cycle, and we now have 4 copies of the DNA that we started with. But just like we saw before, each of these copies are not exactly like what we started with, and they're not exactly like the target DNA that we are trying to generate. So we'll now move into the third cycle of PCR. And I'm going to show you what it looks like when these 4 products then undergo the denaturing or the separation, which is induced by 95 degrees Celsius. And here we see those 4 products separated into their single strands. The primers are going to recognize their targets and anneal through complementary base pairing. This is 1/2 of the pieces of DNA interacting with the primers. And then these are the primers that bind to the other half of the DNA fragments. DNA polymerase would then extend all of these fragments. And now we are at the end of the 3rd cycle of PCR, and we have now generated 8 copies of the DNA that we started with. 2 to the 3rd equals 8, and we can see those 8 fragments that are there. And if we look at these products at the end of this 3rd cycle, we can notice that we do actually have an exact copy of the region that we were targeting for amplification. So this double-stranded DNA molecule is exactly the sequence that was indicated when we first started by the yellow highlighting. And if we look at the other fragments that are generated here, we see that there is another product, which is the exact copy of our target region. So after this 3rd cycle, we have started the process of generating these fragments that are exactly the size of the target that we intended to amplify. And once that we move on to the 4th cycle, which I'm not going to show you because it gets a little bit complicated, but you can diagram this out on a piece of paper, you can see that for each of these exact copies that we have generated, they will each serve as substrates for the DNA polymerase, and they will generate more exact copies of this target region. Sometimes when we study PCR, we can see that the results of a PCR reaction are plotted on a graph. And there's a link here to a website which goes into more detail about this, which I would encourage you to look at, and you can kind of step through this graph to better understand the point that it's trying to illustrate. But if we look at this graph, we can see that along the x-axis we have the number of cycles, and along the y-axis we have the number of copies that have been generated. You'll notice that the scale on the y-axis is a logarithmic scale, and that's because, as you'll see in a minute or two, at the end of, say, 30 cycles of PCR, we're going to generate many, 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 many copies of DNA. So this graph represents what happens at the end of the 3rd cycle, which is what we just talked about. We have a total of 8 DNA copies, and that is going to be shown by the red line on this graph. And we have generated, as we saw in the last slide, 2 exact target copies, and that's shown by the little blue dot on this graph. Note that in cycle 1 or cycle 2, we did not have any exact target copies, so there is no blue dot on this graph because the number of copies was actually 0. So this is one way to kind of keep track of what is happening throughout a PCR amplification cycle. So if we look at, for example, what this graph might look like after, say, 7 cycles, and that's what's shown here, we see that there is an increase in the number of DNA copies. So we now have 2 to the 7th power would be 128 DNA copies. Now we see that the majority of those copies, 114 of them, are exact copies of the target sequence. So as we generate more exact copies of the target sequence, those copies are then going to serve as templates for the next cycle of PCR, and we're going to generate more and more and more of the exact target sequence that is the intended result of PCR amplification. If we come back after 30 cycles, you can see that we would have generated over 1 billion copies of the target sequence. And this is kind of the magic of PCR, is that in a couple hours, sometimes in significantly less than an hour, depending upon the machine that you're using, you can generate 1 billion copies of any fragment of DNA that you're interested in studying. And it's this ability to generate so much DNA so quickly that allows a lot of techniques in biology to be more readily accomplished than they would otherwise. So this is a summary of the first couple of cycles of PCR from your textbook. And one of the things I would definitely encourage you to do as you're learning this material is to sketch this out on your own. It's, again, really helpful to be able to draw this from memory, and be very attentive to the ends of the nucleic acids as you're doing that, because that's going to help orient you. So you have a region that you want to copy. The first step is to use heat to separate the strands or denature the strands. You decrease the temperature a little bit to allow the primers to anneal. And then DNA synthesis proceeds at a temperature that's appropriate for the DNA polymerase, which is usually around 72 degrees. And that's your first cycle. So step A, B, and C are the first cycle. And then that cycle is repeated so that you denature, anneal, elongate, and that's repeated once again, so that at the end of this, you're going to get many, many copies of the piece of DNA that you're interested in amplifying. So one of the things that we expect you to be able to do, in addition to kind of understand all the steps that are involved in PCR, is to be able to generate primers that would be the appropriate sequence for amplifying a particular piece of DNA. So here I have diagrammed for you a segment of DNA, and the question that we're asking is, we would like for you to write down 2 primers that would be required to amplify this sequence of DNA. And one of the things to note is that the only way that this process can work is if you have, in fact, 2 primers, because each primer is required to demarcate the end of the sequence that you're interested in amplifying. So take a minute, you can push pause on your video and write down what you think the sequence of these primers might be, and then you can come back and check the answer. So the answer in this case is one of the primers would have the sequence 5' CTG AG CCC. And that primer would be complementary to the left side of the top strand. And the other primer would be 5' AA GCC TTT. And that primer is complementary to the bottom strand on the right side of this image. So if we imagine separating the 2 strands that are shown above from each other, then we can see how these 2 primers would be annealing to those strands and providing free 3' ends across from template that we want to copy. So this is something that you can practice on your own, and you have to be very attentive to the ends of the DNA molecules and the ends of the primers, and make sure that you're generating complementary and anti-parallel sequences. Once a PCR product is generated, once a PCR reaction is done, you have to think about how are we going to see the results of this reaction. So we know that we're generating 1 billion copies of DNA, But when you're actually doing this, what you do is you take a little tube and you put in your template, your primers, your Taq polymerase, and your deoxynucleotides. So then now you have a tube with some liquid solution in it. After you do the amplification process, you end up with a tube that has 1 billion pieces of DNA in it, but DNA is still really super duper small, and you can't see that. All you see at the end of your reaction is still a tube with some liquid in it. So the technique that is commonly used to be able to visualize the results of a PCR product is something called a gel. And the material that's used to make this gel is called agarose. And you might have seen these before. They typically look something like this. And when you apply your samples to the agarose gel, you still can't see the DNA. In this image, you'll see there's kind of a blue line and a purple line. Those are dyes that are added. Those are actually not the DNA fragments. In order to see the DNA, you have to take that gel and expose it to UV light. And under those conditions, you can actually then visualize the pieces of DNA that have been amplified. So we're going to talk a little bit more about how this gel system works so that you have some understanding of that. So in this image, you can see on the bottom, a researcher is adding samples to a DNA gel. That gel is submerged in a buffer. And on the top part of this image, I've kind of drawn a cartoon of what that gel might look like. At the top of the gel are little holes, basically, that we refer to as wells. And wells are the location to which the sample is applied. The sample is going to be a sample of DNA. And you might remember from the structure of DNA that DNA is actually a charged molecule. If you look at this basic representation of DNA, you can see a bunch of phosphates. Phosphate groups are all negatively charged. So DNA has a net negative charge. So if you, again, look at that image of the gel on the left-hand side, you can see little electrodes that are present. So the way that the gel works is to apply a negative charge to the top of the gel and a positive charge to the bottom of the gel. And that's going to pull the DNA through that gel system. The gel is made up of a molecule called agarose, and agarose is a matrix, and this is actually a microscope image of what that matrix looks like. And what happens is that all the DNA fragments that you have in your solution might be different sizes from each other. And because agarose is a matrix, the smaller the piece of DNA is, the more quickly it's going to move through that matrix, because the small pieces can move through those little holes, but the larger pieces get kind of slowed down by that. So by applying electricity to your gel, you will separate the fragments of DNA so that the large fragments remain near the top and the smaller fragments go towards the bottom of the gel. Now, when you apply your DNA sample, at the end, you're going, and you can visualize your gel. At the end, what you will see are things that we refer to as bands. And bands are the population of DNA fragments that are all the same size. So on this cartoon representation, we have 3 different bands. The top band would have the largest size, and the lowest band would have the smallest size. And size here is referring to the length of the DNA, the number of base pairs that are present in the double-stranded DNA fragment. Another term that's used when we talk about gels is lanes. And lanes are the result of electrophoresis in one particular gel. So a lane may have a single band, it may have no bands, or it may have multiple bands. In order to visualize the DNA in and of itself is not visual. You can't see it. And the way that you can see DNA is by adding a chemical that specifically binds to DNA. And this chemical, one that's commonly used, is called ethynium bromide. And its chemical structure is shown here, and you don't need to memorize that or anything. But what's important to note is that it has the ability to kind of bind in between the bases of double-stranded DNA. And we call that type of binding intercalation because it's kind of wedging itself in between the ladder of the DNA backbone. So it binds really tightly to DNA. In addition to binding to DNA, ethynium bromide is also known to be a fluorescent molecule. Fluorescent means that it will absorb light of a particular wavelength. In this case, that's represented by the blue trace on this graph. So if you expose ethynium bromide to light of around 300 nanometers, it will absorb that light and it will emit that light at a different wavelength. And that's represented here by the red graph. So it will emit light around 620 nanometers, and that light is actually visible to us, and we can capture that by using photographic methods. So ethynium bromide binds specifically to DNA, and it allows us to visualize it by its fluorescence. So at the end of a PCR reaction, you might take your reactions, apply them to an agarose gel to separate the fragments based on size, expose that gel to ultraviolet light in the presence of ethynium bromide, and you might see something that looks like what's shown here. So here we have 5 different lanes. The first lane, labeled number 1, actually has multiple fragments. And those fragments are known sizes of DNA that we've typically purchased from a vendor. And we can label the sizes of those because we know them already based on the manufacturer's specifications. So we can see multiple bands here, the smallest band being towards the bottom, 100 base pairs long, and the larger bands being up towards the top. In lanes 2, 3, 4, and 5, we have the result of PCR amplification, and if I were looking at this gel, I would be able to say that the PCR product that we have generated is something on the order of, say, 275 base pairs long. That's because the fragment that we see is a little bit less than 300 base pairs and a little bit more than 200 base pairs. So when you're doing this type of agarose gel electrophoresis, you're always comparing the size of your product to a known standard, in this case, what we see in lane number 1. So it's by taking your PCR products and applying them to a gel that you can determine that your reaction was successful and what the size of the product that you generated actually is. So as I mentioned, PCR can be used for lots of different applications throughout biology and beyond. And we will be doing PCR in the context of this lab. When we study fruit flies, we're going to use PCR to determine which gene, which alleles a particular fruit fly has. So that's a type of genetic research. You can also, let's say that you're getting your DNA analyzed by something like 23andMe or Ancestry.com. When you spit in a tube and send your DNA off to them to be analyzed, the first thing they do is use PCR to make lots and lots of copies of that DNA, and that's a type of consumer genomics. PCR is not utilized just for kind of genetic techniques. It's also used in evolutionary biology to understand, for example, phylogenetics. Because organisms are related to each other through the process of evolution, we can understand how organisms are different from each other by looking at particular DNA sequences. And this has been a really important tool in terms of understanding evolutionary history by using PCR to copy particular regions of the genome and then study that sequence. So we'll see lots of examples of where PCR is used, but for now, I want you to focus on understanding the basic mechanism that is involved in how PCR works and being able to make predictions about what primers might be used to amplify a certain sequence. And we will pick up on this later.